In this part of the methods review, we will go over two of the key non-invasive methods used in modern neurolinguistics research: electro and magnetoencephalography. Electroencephalography, or EEG, is the recording of electrical fields that are generated by brain activity using an array of electrodes that are placed on the scalp of the participant. Electrical activity in the brain generates magnetic fields. Magnetoencephalography, or MEG, is the recording of magnetic fields that are generated simultaneously with electric fields using sensors placed around the scalp. As MEG sensors are at an even greater distance from the neurons that generate the signal than EEG electrodes, this method is more complicated and technically demanding than EEG. Data collection for MEG requires a magnetically shielded room and highly sensitive detectors. These are called superconductive quantum interference devices, or SQUIDs. The physical premise of EEG and MEG is the electrical field that is generated by postsynaptic activity of large populations of neurons, specifically the relatively large pyramidal cells in the cortex. At the same time, magnetic fields are generated simultaneously around the axis of each neuron. Such activity, when generated simultaneously by tens of thousands of neurons, can be detected from a distance. The resultant local field potentials, or LFPs, and local magnetic fields, or LMFs, are the building blocks of EEG and MEG signals. Recording of electric potentials and magnetic fields from a distance from their source and the interpretation of such recordings are associated with two key problems: the forward and the inverse problem. The forward problem is the calculation of the scalp electric field caused by neuronal sources. It is a solvable problem, and its solution is somewhat easier for MEG than for EEG. This is because the MEG signals are less affected by tissues between the brain and the scalp than EEG. The inverse problem, the second problem, is the estimation of what specific neuronal sources in the brain generated the fields that were recorded from the scalp or from the MEG sensors. Unlike the forward problem, which is solvable, the reverse problem is a so-called ill-posed one. It means that it has an unlimited number of solutions. A common approach to this problem is to make assumptions about sources. For example, auditory responses can be fairly safely presumed to originate from anatomically defined auditory cortex. The hypothetical source of the observed potentials of fields in the brain is called equivalent current dipole. EEG electrodes and MEG sensors are often referred to as channels. So how many channels does one need to get adequate sampling of EEG or MEG signals in space? For EEG, 21 channels routinely used in clinical practice is not sufficient for research. Instead, 128 channels are used. So-called dense array EEG systems use as many as 256 channels. For MEG, whole head systems usually come with somewhere between 200 and 300 sensors. A useful computational approach for dealing with such a large number of channels is called independent component analysis, or ICA. Basically, it reduces the number of channels to a minimum number of signals that are statistically independent. This is quite helpful for extracting biologically relevant information as well as identifying and discarding artifacts. EEG and MEG offer several advantages over another common neuroimaging technique, functional magnetic resonance imaging. Signals recorded by EEG and MEG are direct measurements of neural activity, unlike changes in brain metabolism that is captured by fMRI. Also, both EEG and MEG have a high resolution on the order of milliseconds, which allows for measurement of precise timing in neural events, as well as functional and effective connectivity between different brain regions. Finally, functional neuroimaging studies are associated with scanner noise, which can be quite loud. Experiments performed with EEG or MEG systems do not have that problem. The data can be collected in silence, and these methods thus are particularly well suited for studies of hearing, speech, or language. Orientations of neurons within cortex and the orientations of electric and magnetic fields generated by these neurons leads to differences in sensitivity of EEG and MEG to the spatial orientation of the sources. These differences are exemplified in this figure by density of EEG and MEG sources of alpha and mu oscillations in the brain. You can see that EEG is sensitive to sources originating both from cortical sulci and gyri. These are called tangential and radial sources, respectively, whereas MEG is sensitive to tangential but not radial sources. MEG is selective to activity in cortical sulci, but also it has relatively better spatial resolution than EEG. Studying interaction between signals recorded from different EEG or MEG channels allows to make inferences about connectivity between different brain regions. Functional connectivity is the statistical association between two signals. It can be measured using analysis methods such as phase locking value. Two signals that have a consistent phase relationship over time in a particular frequency band or a high phase locking value are said to represent functional connectivity. On the other hand, effective connectivity is the causal influence of one system or a brain site on another. Methods of analysis of effective connectivity include dynamic causal modeling and Granger causality. Connectivity between two locations in the brain can be measured by examining phase and amplitude relationships of recorded signals across frequencies. This is called cross-frequency coupling. As illustrated in this schematic, there are several ways two locations in the brain can communicate with one another. The relationship between sites A and B can be described as power-to-power coupling. Here, power or magnitude of the signal changes in a consistent way, but in different frequencies. Phase-to-phase coupling, as exemplified by sites A and C, is when a certain phase of the signal in A is associated with the same phase in C over time. Phase-to-power or phase-amplitude coupling, exemplified by sites A and D, is when the phase of a lower frequency oscillation in site A shapes the amplitude of a higher frequency oscillation in D. By analogy, other types of coupling include phase frequency, power frequency, and frequency. Speaking of frequencies, EEG or MEG oscillations occur at a range of different frequencies. The conventional bands include delta, theta, alpha, beta, gamma, and high gamma. However, the latter, the high gamma band, may be difficult to detect using MEG or MEG due to its low magnitude in the EEG or MEG signal. This example shows the power spectrum of the human EEG signal at rest. One can appreciate the overall decrease in power with frequency, as well as a characteristic peak that corresponds to the alpha frequency band. Signal magnitude in each frequency band reflects the extent of activation or the number of neurons, as well as the degree of synchrony between them. It is important to distinguish evoked and induced activity that is reflected in the EEG or MEG signal. Evoked responses are both phased and time-locked to the stimulus. Phase locking means that the peaks of responses occur at the same time from one presentation of the stimulus or trial to another. Thus, averaging multiple trials can enhance the evoked response and suppress other components that are considered noise, as shown in the left panel of this figure. The amplitude of the averaged evoked response will depend on the amplitude of the single trial responses and their synchrony or phase locking. In contrast, induced EEG or MEG response occurs after the stimulus but has no strict phase relationship with it. Such responses represent modulation of ongoing brain activity by the stimulus. As shown in the middle panel of the figure, simple across-trial averaging that worked well for evoked responses doesn't work for induced activity, as the single-trial waveforms eventually cancel each other out. Therefore, nonlinear analysis methods, such as time-frequency decomposition, as shown in the right panel, must be applied before averaging for analysis of such responses. In conclusion, EEG and MEG are non-invasive brain methods that offer a direct measure of neural activity with good spatiotemporal resolution. To compare EEG and MEG, the EEG is a widely available, portable, relatively inexpensive, and well-understood method. On the other hand, the MEG is somewhat less available, less portable, and more costly, but it offers better sensitivity to neural activity that originates from brain sulci, including human auditory cortex. Both methods have made and continue to make important contributions to our understanding of hearing, speech, and language processing.